Thank you all for coming today. I'm going to be giving a review of our RNA-seq portfolio and also presenting some data from our internal scientists as well as a few customer publications showing the array of applications that are enabled with our RNA-seq products. So we offer RNA-seq products that cover an array of sample inputs and sample types, from high-quality samples down to very degraded samples such as laser capture and archived samples. And they enable research in a wide variety of research areas, including pest resistance and drought resistance, global transcriptomics, tissue-specific gene expression, and development. While RNA-seq can provide a great deal of information about your sample types, there's also many challenges in preparing samples and generating data, such as limited or degraded samples, PCR bias or not enough data from your sequencing results, as well as inaccurate quantification and inaccurate mapping. And our systems provide a range of solutions to each of these common challenges, including compatibility with low inputs and low quality samples. We have PCR-free workflows and depletion of unwanted transcripts to enrich the amount of information you get from your data, as well as molecular tags and strand information. So this is a brief overview of our RNA-seq portfolio. Our portfolio includes 2 complete RNA-seq library systems. These are strand-specific RNA-seq libraries that feature a targeted depletion technology that can be customized to any transcript, particularly our RNA, and also highly expressed genes such as housekeeping genes. We also offer our RNA-seq V2 system, which is compatible with a range of downstream workflows and DNA library prep kits from many vendors, including our own. And finally, we also offer a product for target enrichment for RNA, which JD briefly mentioned, and this can be used to study translocations, gene fusions, and viral integration sites. So I'll be focusing today on our universal solo and RNA-seq V2 systems and showing data from each of these kits. All of these kits, again, are designed to work with both high quality and degraded samples, and together they cover a wide range of inputs from 100 nanograms down to a single cell. I'll start with our universal and solo RNA-seq systems. So these are both complete stranded library systems that feature our InDA-C technology, which I'll describe in detail in a moment, which can be customized to any organism and any transcript. Universal is for everyday samples, higher inputs, and solo is for very low input or degraded samples. Sorry. Okay. So our InDA-C insert-dependent adapter cleavage technology, again, it can be customized for any transcript. And the way the system works is you prepare libraries. These are Illumina-compatible libraries, and then apply the customized probes, and they're specific to the sequences of interest, and they're tiled across the sequences. We can vary this for degraded samples so that even with a degraded sample, you can still get very good reduction of unwanted transcripts. And then these probes facilitate degradation of these sequences so that only desired sequences or potentially informative sequences remain in the final library. And because this design is specific to each transcript, you have minimal off-target effects, and again, it's customizable to any organism. So this is some sample data from each of these 2 kits. So in Universal, we have RNA from several different species. These are all of our catalog kits that we have available. So shown here at the bottom is the design that's in the depletion for each. So for Arabidopsis here, we have a design to cytoplasmic rRNA, as well as a design to the chloroplast rRNA. And you see that before applying the InDA-C, the rRNA reads comprise about 40% of all of the reads in the library. And after targeted depletion, only about 2% of these sequences remain. And this depletion is robust across all of our species for universal. In the solo system, using 100 picogram inputs in this test data, you see rRNA up to 80% of the libraries, and with depletion, we see about an 80% depletion. So shown here is an experiment showing the gene expression. In this, we looked at the difference between a human brain sample and a human reference, a human universal reference RNA, and looked at the global gene expression changes and compared the depleted and non-depleted samples. And in both of these samples, you see a very high correlation between the non-depleted and the depleted samples. We also compared the differential expression of the two samples that I just mentioned to a qPCR gene expression panel of 400 genes. And again, these are only depleted libraries in the RNA-seq data this time, and then TACMAN expression for the same two samples. And again, we see very high correlation, showing that we're seeing minimal off-target and minimal impact on the gene expression, the global gene expression of these samples with the targeted depletion. Shown here is an experiment looking at the targeted depletion in the design of rRNA and globin. And before depletion, these samples have very relatively few informative reads, anywhere from 25 to 30% of the sample. With depletion, you have about a threefold increase in the amount of informative reads. So about 90% of the data now is comprised of potentially informative reads rather than abundant rRNA and globin that's minimally informative. Now I'd like to talk about the solo system in a little bit more detail. This system is for lower input samples as well as degraded material, anywhere from 10 picogram to 10 nanogram of total RNA, or 1 to 500 cells, and we include a direct cell lysis workflow in this kit, so cells can be sorted or used from laser capture directly into the buffer and processed through the kit. And again, this kit also has the customizable depletion for any organism, and shown here are examples of several of the designs that we've already created: goat, pig, cow, grape, and corn. And this kit also features a molecular tag after the barcode for duplicate molecule identification. And here's some sample data from that kit looking at inputs over a wide range. So on the top left here, so for all of these graphs, we're starting at 1,000 cells on the left and then going down to a single cell of input. On the top left figure here, we see robust rRNA reduction of down to 10% or less of reads across all of these inputs. We see strand specificity of greater than 90% across the range of inputs. Consistent read distribution across the whole transcriptome for each of these inputs. And then gene detection consistent with input level, meaning that we see consistent detection of genes across the first three inputs, but then at the single cell, you do see a drop because not every gene in the transcriptome is expressed in each individual cell. And as I mentioned, this kit is also compatible with degraded samples, and shown here is an experiment with some severely degraded RNA with RIN values of 2.1 to 2.6 and a DV200 of 10 to 20%. For anybody not familiar with this metric, It's basically a metric of the proportion of your sample that's 200 base pairs or longer, and is an indicator of molecules in your RNA that will make it into the final library. So even with some samples that are quite terrible looking, we're able to generate a library. And the experiment here used plasma RNA and compared to a whole cell RNA sample. And what we see here is detection of a wide range of RNA species in addition to mRNA, which is consistent with this sample type. And this is an overview of those two systems. So these systems are customizable and highly flexible with our higher input universal system and the low and degraded input solo system. They can be used with the probe sets for both kits as well as custom. And these kits are, the probe sets are actually optional as well. So it's flexible for a wide range of experiments. And again, here are a number of example designs that we've already created for this system, showing the breadth of organisms that can be covered. So we've done designs for soybean, rice, Aspergillus mold, Arabidopsis, Plumeria, which is another plant pathogen, grapes, corn, and Venus flytrap. Next, I'll go into our RNA-seq V2 system in a little bit more detail. This features a single primer isothermal amplification technology, which is a non-PCR based amplification technology that better preserves the stoichiometry of the sample. This provides robust cDNA amplification for multiple workflows, and it's compatible with a range of input, 500 picograms to 100 nanograms, with both high quality and degraded samples. So with this technology, you can start with high quality and degraded samples, lower inputs, and both total and poly-A RNA. And the cDNA is created and amplified with the single primer technology. So again, this means that the stoichiometry of the sample is better preserved because it's a non-PCR amplification. And then the product can be used with many downstream workflows, so not only library prep and sequencing, but also qPCR and sample archival. And with this technology, we see even distribution of the reads across sequencing. So on the top is a highly expressed housekeeping gene in 2 different samples, and the bottom is a gene that's more low expression, and we see even distribution, even coverage of these genes across the entire transcript for both high and low expressed genes. And as I mentioned earlier, this kit can be paired with many different DNA library prep kits, including our own. And our DNA library prep kits feature a dimer-free technology, so it's an optimized adapter ligation chemistry that has fewer dimers in the final libraries than our competitors. And you can see here across a range of inputs from 1 nanogram all the way down to 10 picograms, so a single cell input, we have minimal adapter dimer, giving you more informative reads in your data. And here's some more data showing the RNA-seq V2 product paired with our DNA library systems. This is with our rapid library system, and together these kits can be used to go from RNA to Illumina compatible library in a single day. So we see high concordance of samples across a range of inputs. This is a higher input, so a PCR-free workflow with the rapid library system from 100 nanograms to 750 nanograms. And then it also gives you high confidence gene expression data. Again, I mentioned you have the preservation of stoichiometry in your sample, and this again is a comparison of 2 samples, differential gene expression with both RNA-seq as well as TaqMan, and you see high concordance in gene expression changes from both low and high expressed samples, or low and high changes in gene expression, sorry. So now I'm going to go through a few case studies of customer publications featuring our kits. So the first study looked at daytime soybean transcriptome fluctuations, so global gene expression during water deficit stress, and they looked at multiple time points throughout a 24-hour period. In this study, they identified nearly 5,000 genes associated with water stress in soybean, and they linked these gene expression and changes 2 different times of the day, with stress-related pathways most affected during the middle of the day. And in the figure on the left, it's a little bit difficult to see, but this is the concordance of the controls across several different time points. So this is ZT0, 4, 8, 12, 16, and 20. And on the right is for the stress-associated genes. And this is with our whole transcriptome stranded library kit, so showing high concordance of samples across the controls. And then at right is a qPCR validation study, and you see good concordance in gene expression for the very low expressed genes between qPCR and RNA-seq across each of the time points. And here's a study showing discovery of non-coding RNAs in barley. This group performed whole transcriptome RNA-seq with our RNA-seq V2 system and identified 62 circular RNAs in barley seed transfer cells. So these RNAs are much more lowly expressed than an mRNA, and they still had robust detection of these RNAs with our kit. And they looked at the gene expression changes in these circular RNAs across tissues and in response to micronutrient changes. Again, this is a bit difficult to see, but we're seeing good concordance of the controls across different micronutrient tests compared to samples where the nutrients have been changed. And the last case study that I'll review is a study of, again, non-coding RNA discovery in Dictyostelium, which is an amoeba found in the soil. And in this study, they performed whole transcriptome RNA-seq to identify link RNAs and other non-poly-A RNA. And they identified hundreds of intergenic link RNA loci and characterized the temporal expression pattern of these, as well as their correlation between link RNA and mRNA in different points in development. And shown here in the chromosome map, they detect the link RNA loci across the entire chromosome. And in the figures on the right, they compare the expression of these link RNAs as well as the abundance of transcript, the length, and the GC content to mRNAs. So the link RNAs are much less abundant than mRNA, and they're still reliably detected with our kit. And then the other one of interest here is that the GC content is much lower in the link RNAs, only about 20% versus upwards of 30% in the mRNA, and they still have consistent detection of those. So in conclusion, I've reviewed our universal and solo RNA-seq systems. Again, these are both stranded library prep systems for whole transcriptome RNA-seq, and they feature our targeted depletion technology, and our RNA-seq V2 system for robust cDNA generation compatible with a range of downstream applications. In addition, I didn't really talk about these today, but we do also offer a range of kits for DNA library preparation, as well as whole genome and RBS methyl-seq, and also our target enrichment systems that JD and David reviewed. Thank you for your time. Any questions?
Are you familiar with any strategy to enrich mRNA transcripts that are specifically loaded onto ribosomes and being translated?
So we have a couple of customers that are using that, and with that they're using, I believe, our solo system, and they're doing a pull down up front, And then they've done some custom designs to remove anything else that's abundant in their sample to get better detection of those transcripts.
Does that work well?
It's working. I haven't seen any of the final data yet, but we do have a couple of customers. I'd be happy to give you my card and see if you would like to discuss them and learn a little bit more about their methods.
Thank you.
Thank you.